So welcome to demonstration video 1, where we're looking at this setup from the experiment and cluster quantification of this cow FMD infection RNA-seq study. So that's what we're going to focus on now. So this study was published 2 years ago. It was looking at the immune response associated with foot-and-mouth disease virus infection in cattle. To retrieve the data for this study, we were able to go to get the bioproject ID, go to the SRA with a query to get the corresponding file names, go to the SRA one selectors to extract the accession IDs, and then we could use FastQDump and through the SRA toolkit to actually download the files itself. We're dealing with 15 samples, all of which were taken from the ventral soft palate tissue of these cattle. There's 5 each from 3 breeds, hence 15. 2 of those breeds are Bos indicus. They correspond to the names Malna Gita and Hadikar. The 3rd breed is Holstein Friesian, which is Bos taurus. I guess the question is the immune response higher in bull cows with the if they have a milder response to FMDV serotype O India or 275 infection? And the data quality is quite good. It's dealing with transcriptome sequencing of using the Illumina HiSeq 2500 platform with 151 base pair reads. So the data that we're looking at can be summarized in this manner, that we have the breed effect and then the treatment effect. So the breed effect, we have a Malna Gita, a Hadikar, and the Holstein Friesian groups. The treatment then is FMDV infection for 3 of each of the 3 breeds. And then those 2 that are unaffected for each of the 3 breeds as well. So hence we have 9 versus 6 for treatment and 3 by 3 for 5 by 3 for the breeds. Then of course we have the error associated with other factors. So thus we get this kind of simple model where we have our description level for a transcript. We have our breed effect, our treatment effect, and then some sort of error we have to consider. So the suggestion for this practical is that you might examine 5 samples for one breed, because analyzing 15 might be a lot. And this is so you have enough time to finish the lab, because dealing with this, the size of these samples will take a little bit of time. First, we'll quantify them with Callisto, and then we'll assess that expression levels with Sleuth. So for Sleuth, we'll have a table like this where we have the accession numbers that are of interest. There's 5 here. We have the condition here of the infection status. Then we have the breed, which is arbitrarily Malna Gita for this arbitrary set of 5. And then we have the file extensions where they're located, where you change you to your directory. So what you do first is copy data from this folder. So you might symlink the FASTQ files over because they take up a lot of space. So linking them locally, and the final dot at the end indicates that you're putting them in your current working directory. So you need to set up a directory for this. What you do next is run fastqc on all of them. That will generate a HTML file and a zip file containing the quality score information. You will also need to copy other items for the FastX quality trimmer, which we'll talk about shortly. FastP, which will Callisto, which we'll use later, and then FastP as well, which follows the FastQ quality trimmer step. We'll also map our reads using Callisto to the Bos taurus cDNA set from the reference genome. So that's our step. So after FastQC, we go to this FastQ quality trimmer. So we use that to trim reads that are low quality or are short. So here we have a -T30, which means that we keep bases only quality score, Fred scaled quality score value of at least 30 and a length of at least 120 in this example here. But you can toggle that to something more stringent if you want as well. This is our input file name, which you will have to change, and then our output file name. You'll need to do this for the forward reads and reverse reads, _1 and _2, as you saw before. We have _1 and then _2. And you can add some verbosity there, so you can send that to output as some sort of file containing the results of your trimming. What we'll find then later is that you'll remove some reads, but relatively few of them. Not too many per sample, and you'll see if you summarize across the different samples, you can see that most of them, there are different numbers for each particular one, but not too many are removed, indicating that the quality is good generally, which is reassuring. FastP is a very good tool, multifunctional tool. It can handle long or short, single or pared down reads. It filters low quality reads again, in addition to the FastQ quality trimmer. It can trim reads as well. It removes adapters. It can remove poly A, G, X tracks as well. We can view that with a HTML file, which we'll do with multi-QC later. And you can also get more information on the exact adapters that are removed at this GitHub page. So to run that, what we can do is add in the overrepresentation analysis toggle. We can set it to HTML. We have our input file and our output file. So for each sample forward and reverse, we'll need to do each forward and reverse file individually. So that means you'll do it 10 times in effect. And then if you want to install multi-QC to collate these HTML files that we've generated from FastQ at the start, and at the end, we can use pip to install that. So we pip install multi-QC, and then it should be executable from place. If not, you can execute it from this file stem as well. You can copy that over with this command as well. And what you'll do is just do multi-QC in your current directory, and that will generate a HTML report in your current working directory, collating all the HTML files that are there and of interest. What you'll find is that you'll look at these things like parent-based sequence quality, the n-based content, the per tile sequence quality, the quality scores, GC content, sequence length distribution, the duplication levels, no representation, analysis, adapters, and k-mers. So there's quite a few items there. So let's try some of that on the server now. So you may also want to use the commands in the Word document for this lab as well, which makes things more easy. So the first thing you want to do here is zoom in a bit, make a directory, let's call it RNA_seq, go to that directory. What we're going to do is copy over that data we have and check that it actually worked. Sometimes you have issues. So let's see if those worked. Yeah, we see those 10 files are copied over. And then we can copy over those things in batch. The FastX quality trimmer. Oops, I just copied the wrong thing. The FastX quality trimmer. Yeah, we have it there. Callisto and FastP. So let's just check that we have these things. So we just need to check that everything is copied over. We see there that we should have everything nearly now. So FastP, the FastX quality trimmer, and then Callisto as well. So that means now we can take each of these files and run FastQC on them. So if we look at the first one, 398, we can just run through FastQC. It'll say start analysis of this particular one, and then it'll iterate through each few percent, like 5%, 10%, 15%, and so on and so on. It may take some time. So what you can do is a handy trick is to do Control Z, put it in the background, and then for the next one, you can actually send this to. For the next one, you can send this to output. So you can send that to the corresponding file extension, that out, and then run behind. Check your jobs. There's 2 jobs running, and you should be able to see your output in this file there that it's just started. And then be able to do that for each of the different files that are present. So that might take some time, but it's always valuable to do proper QC of your different files. So I'll let you proceed with that. And you can see that it takes a bit of time to run. But then at the end, we'll have generated some HTML files and also zip files that we can examine later with multi-QC. So you can look at the FastQ files yourself, but we'll mainly collate them together for multi-QC later. What we'll do at the end of when this is run is take our FastQ file and run the FastX quality trimmer on that. So then if we look at our files for that particular sample, be it we have a HTML file and a zip file that is generated by FastQC. We should be able to see them then for this particular sample. If you look at FastQ file 1, you see there we have our FastQC one there. It's called HTML. And if we do want to view that individual one, we can just use Google Chrome to view that one on the server. So you see there we have the basic information you should be familiar with in terms of the FastQC output, which is generally quite good as you scan through. And we'll collect that later for multi-QC. What we should do next then is the FastX quality trimmer. So for that, we use the command that we had before that you can take from the worksheet that we have. Oh dear. So what we're trying to do there is put in our corresponding file, our input file. So we change these I for input. So that's SOR 398_1. We have our Z. So we change that to SOR 398_1, and that's our output file with the underscore trim at the end. And then we're just going to send our verbose output to this trim file. So if we then run that, that will take some time to complete. Then we can look at our trimming file at the end of that process. So if we look at our trimming file, we can look at our output .trim. So we can see there that we have other things in the background, but what we're mainly looking at is this text file where we see too many short reads. And if we look at the other ones that correspond as well, you can see that for each of them, about 0% are removed, or maybe 1% in certain instances. So that's kind of what we expect to get for that FastQ quality trimmer. So what you can do is do that for all the other samples. Following that, we wish to run the FastQ over-representation analysis. And again, the structure for that is the same. So we take our sample that's of interest. And here we're going to put in our input file. So we're going to set the same file, stem 398. That's the HTML output file, I should say. And our input file is next 398_1_trim. So we have to do this twice for each pair of files corresponding to one sample. So it's 10 in total. And then we got _1.trim.fastp. That's our output file at the end. So we run that, we'll find that we are able to look at this HTML file for that particular sample. So let's look at that next. So what we can do is look at that fastp HTML file. So if we do that, we can view that in Chrome. We can even zoom in with Control++ to see it more easily. So you can see there we have our reads listed as single end here, as far as me, but it should be a paired end. The duplication rate is relatively high because the samples have been amplified a lot, presumably. We see that the difference between before total bases before and after is some very small difference. So it's not too bad. And you see then our quality scores across the reads. The first few bases are poor, up to maybe base. The first 5 bases are poor, and then the slight dip here in the middle, and then slightly deteriorates here, but the quality scores are generally very, very high, which is good to see. So that's kind of reassuring, and we can summarize all this then with multi-QC, which we can use next after this FastP step. What we need to do next, however, is use FastQ to verify these files. So what we can do is just take our, if we take that sample again, we're just going to run FastQ on our final samples, And you do this for all 10, underscore trim, underscore fast p, so we have it named appropriately on the FASTQ file like that. So if we run that, that again, we'll read that in and we'll generate HTML and zip file again for that file. And as mentioned, you can repeat that for all the different files, and we'll then collate that into a multi-QC file in due course. So to run multi-QC, we can use the command in our worksheet again. I suggest one here is just to collate for all the SOR related HTML files, make an interactive QC report with that title there. So we can make it as effectively as HTML and as PDF if we want. There's different options. So you can see there what it's collating through there is all the different files, scanning all the files to get the ones that have HTML links. It says it found 19 reports and it's generated this one. So if we open Google.-Chrome and get this report, then we can view that with Google Chrome. So you can see there we have our kind of before and after of our different files present, and you can use this button here to toggle to view as much as we feasibly can inside the screen. So you can see there for the top one, for example, the duplication is slightly reduced after QC, the GC is slightly fallen, number of sequences is slightly changed. So it's nothing too drastic, which is reassuring here. That's the general steps. In terms of the sequence counts, you kind of see before and after for the different ones again. Sequence quality histograms, again, is very, very good before and even better after. So that's reassuring. The quality scores, yeah, they're very good. The GC content is a bit of a puzzle that it's such a wide variation, but there's differences in expression here. And we have to remember that we're sampling different transcripts at a point in time. So that would suggest that we likely need a lot of different samples from these animals to really understand their variation in the immune response. So you see the duplication levels are low, the overrepresented sequences are somewhat as you would expect for these kind of libraries, and the same for the adapter content. It varies quite widely, and we see then that we've more green segments for the after sequencing and before sequencing, thanks to fast P. So that's a short version of the QC results. So that's that part kind of completed. So now that gives us an opportunity to move on to the Callisto analysis next. So for this Callisto part, we're using this QC FASTQ files to quantify the abundances from our RNA-seq using pseudo-alignment. We're looking just at a partial alignment across a set of compatible k-mers, as mentioned in the introductory videos. And then we're able to implement this and use the extra compute time that we've saved using pseudo-alignment to bootstrap the estimates of the transcript levels. So if we imagine that we have different transcripts here in pink, blue, and green, and we have a read in black, here we can map a read to these different isoforms, where we're looking at the isoforms in terms of their Bruin graph construction as a set of k-mers with k-mer classes. So when you map a read there, the black read to these black nodes here, we don't need all the nodes because nodes V1, V2, V3 are all part of the same k-mer class. So therefore, we can actually map only to V1, V4, and V5, and that will distinguish between the different isoforms that are possible here. So therefore, we're thinking about the reads being assigned isoforms rather than identifying the exact location. That's a second step. But that principle can be used in Callisto. And so what we're going to do here is a few steps. We're firstly going to clone the GitHub page, go to the folder, build that, make a directory called build that, use cd cmake make, and go to the executable path and export your path to add that location to your path, so you can use Callisto from wherever. Or you can just copy over the Callisto as we did earlier, which is probably the easiest option. By doing that, what we'll be able to do is index quantify skip the slide, we'll be able to index our reference cDNAs using K-MERF31. And what we'll see at the end, after all this, is that we are using the ensemble release 110 from the 4th of May 23. This will take a few minutes to run, so bear that in mind. Or you can copy it over to your current working directory. So I suggest the latter. What you will see is this kind of output later when we use it. So let's go to the server next for the quantification process. So there we're using the quant function inside Callisto, where we're taking our index there -I. Our output is called, we have to change the directory name there. We're using 2 threads. We're using 100 bootstraps. And we have our 2 FASTQ files there as input. So we give it that input, we'll get output like this. It's looking at K-MERF31, it's looking at the number of targets present, the number of different K-MERF classes, number of input reads that can be pseudo-aligned, and this will give us our quantification in terms of TPM at the end. So let's switch to the server now. What we can do for efficiency, just copy over our reference index there, BOS2, which is based on the BOS-Taurus cDNA reference genome. And then what we want to do is next run Callisto quant on our file of interest. So we might call that first, use to start with the first sample 398. And we can toggle the threads here to maybe something that really suits us. We're using 100 bootstraps in this instance, and then we need to give it the correct input files that we need. So no, that's going to be trim FastP with FastQ at the end. And again, here we use the same file stem 398_2 trim FastP, and these are not compressed files. So we press enter. And let's see what's going on here. So what we can see is that I need to call my clubbing index BOS2. So I need to call it BOS2. And this process will take a while because there are many reads present. So you'll have to wait a few minutes while all of this runs for the different samples. And then what this will do is generate a directory with this file extension, and you'll be able to examine the reads in that directory. So we can see that our number of bootstraps is now completing for that first sample. So what we can do to test that out is firstly go to that directory. So go to that particular folder, and we see inside it that we have the abundance.tsv file, the run info file, which just contains the setup for this particular run. So you can see there are a number of targets, 35K, the 100 bootstraps, the number of processed and pseudo-aligned reads, the Callisto version, and the actual command used. If we look at the abundance.hi5 file, we can't actually look at it at all. It's not machine readable. But we can look at the TSV file, which contains the target transcript, its length, its effective length, the number of counts, and the TPM. So you can see there for some values it varies and goes on for quite for all the different transcripts that are present. So we got 38K. So we can view this in R. So let's open R, and if we do a quick histogram, we can draw a histogram, hopefully, by reading in this CSV file in our local folder. What I'll have to do is exit this folder, go back to the main folder. So launch a loader for the current R version, launch R. And then what we're doing is then we're reading in a CSV file from that folder, the abundance.tsv file, which is separated by tab. That's reading in a set 1. And they're taking set 1 and we're taking the TPM. We're taking the log of that. And we're having a lot of breaks. So you can see that at fine level, the changes, differences in expression for each particular transcript, and we're adding a grid just for legibility. And if we do that, we'll see something like this, where you can see there the kind of spread of expression values that are present. If we do that without the log, it's not quite as useful. So you can see without the log, it's not because there's such a wide spread, it doesn't really tell us very much. That's why log scaling makes more sense. So next now we can go, we finished this, so we can now go on to using sleuth, which follows up in our next demonstration video. So remember to complete that for all the 10 libraries that you're going to be the 5 samples in total that you're going to examine. And we'll move on to sleuth next.